Good morning, everyone, or good afternoon to some of our colleagues in Germany, given one of our panelists here today from Vivoryon. My name is Jason McCarthy. I'm the head of biotechnology research at Maxim Group. I'm also joined by my colleague, Joanne Lee, from Maxim's biotech team. And I would like to welcome everyone once again to our ongoing virtual series of conferences, panels, and fireside chats hosted by ANVS, Maxim's digital online platform. The content over the past year across all sectors has been exceptional, to say the least, and combined with the end user experience has continued to drive the ANVS platform to new heights. And today that trend is going to continue as we bring everyone back into the always very active biotech and pharma community, this time with an interactive panel discussion on Alzheimer's disease. For any topic really across any sector, timing is everything on the street, right? So why talk about Alzheimer's disease right now today? As everyone in the US is probably starting to think about the long weekend ahead and Memorial Day and that unofficial start of summer, which we really need given what we went through in the last year, but really not so fast. And there is a major event in the Alzheimer's drug development space that is looming, and that's the Biogen aducanumab PDUFA on June 7th. And the questions are going to be, is it going to be an approval? Is it going to be some approval on a narrow label, or is it going to be a CRL? And I guess, really we're all going to find out soon enough. And there are some important topics within the context of this event, from the needs in the space, shifting regulator or regulatory views of Alzheimer's disease drug development, where the amyloid and tau plaque clearance approaches are and where they're going. And most important from my perspective, because of course it's directly related to this ANVS panel discussion, the rapidly evolving drug development space targeting neuroinflammation and neurotoxic cascade, and more or less just upstream of where the plaques or plaque clearance is. So everything that's happened over the past decade, or probably longer, around amyloid, from Lilly's solanezumab, and we all remember that, to their new antibody donanemab, to this whole aducanumab odyssey, up and down and kind of really all around over the past 18 months or so, has brought an enormous amount of knowledge, eye-opening and shifting thinking around Alzheimer's disease, and it's ushered in a wave of companies targeting other areas of the disease. So you could look at what Cassava Sciences drug simufilam did last February on Cognition. You could look at INmune Bio, which demonstrated in early data utility of an MRI white matter-free water as a viable biomarker. We're going to talk about that. Annovis just last week with ANVS 401 also impacted cognition. Cyclo Therapeutics, via all of its work in Niemann-Pick disease, and that's a completely different topic. You'd have to go take a look at it. Really exciting stuff. Found that its drug, Trapsol Cyclo, may also have utility in Alzheimer's. And Vivoryon, with its drug, varoglutamstat, always butcher that. I use the word, it's quote unquote quietly in the EU in a phase 2B study, and it's moving towards a US study, and it's relatively unnoticed in my view. So the bottom line is that Alzheimer's disease has been busy, to say the least, and that activity is likely going to ramp up with the aducanumab PDUFA in June, and of course the AAIC meeting in July, plus all the activity around all these companies that are here with us today. That's going to continue. So before you head out for Memorial Day, I want everyone to leave with this panel on Alzheimer's disease. And as I started this talk, I'll kind of end with it, is that timing really matters in biotech. And right now it's all about Alzheimer's disease. So I'd like to, with that, welcome our panelists, CEO of Annovis Bio, Maria Maccecchini, from Vivoryon Therapeutics, CEO Ulrich Dauer, Remi Barbier, CEO of Cassava Sciences, RJ Tesi, welcome back, from INmune Bio, CEO, and Sharon Hrynkow, the Chief Scientific Officer of Cyclo Therapeutics. And really, before we get started, I just want to set the stage for all of our viewers and have each one of you, if you can just give us a minute or so on your companies and what you're working on in Alzheimer's disease, that would be extremely helpful. And I think the best place to start would be with you, Maria, at Annovis, given all of the activity this company has gone through in just the last week or 10 days or so, that's really propelled that stock to new heights. So Maria, I'll let you please give us an introduction.
Well, thank you so much, Jason. I think this is the greatest panel at the best time because, as Jason just said, we have novel approaches. We went away from removing A beta or plaque, and all these novel approaches really promise hope to whoever has a disease that robs their brain. And there is a really good chance that some of these approaches will be synergistic because there is pretty much not one neurodegenerative, no, I should say degenerative disease, chronic disease, that is cured with one drug. So I'm very happy to be here. I'm the founder and CEO of Annovis Bio, and we have been pushing a way of looking at the brain that does not really differentiate between Alzheimer's, Parkinson's, frontotemporal dementia, or other neurodegenerative diseases. We look at nerve cells as the same, and they do get sick the same way, and they do die the same way. And our drug improves their health. And we had shown that in animals, but now we've shown it in humans, and it protects nerve cells from dying. So what we released a little bit over a month ago is that in Parkinson's patients, we fully recover one test, that is the speed test, that is the coding test. The Parkinson's patients are much faster at coding fields after 25 days of taking our drug than before. In Parkinson's, we also have inflammatory markers that are reduced down to the levels of healthy, normal human beings. In Alzheimer's, we just released the Alzheimer data, and basically we see in the ADAS-Cog11 that we have statistical improvement because patients do a lot better 25 days after. They also do better compared to placebo, but it's not statistically significant. Interestingly enough, in the coding test, that is neither a UPDRS test nor an ADAS-Cog test, we did it for Alzheimer's and also for Parkinson's. And as I was saying, it works beautifully in Parkinson's. And as of yesterday, I know it works beautifully in Alzheimer's. So whether they have Alzheimer's or Parkinson's disease, they code much faster, statistically significant, whether compared to baseline or compared to placebo. And so I want to give the stage now to the other companies that all have great results that really promise that we can solve this problem of neurodegeneration.
Right, and I think I want to jump over to Uli, and I just met Uli and his team relatively recently and just was impressed with what they're doing. And like I said at the beginning, it's quiet, like it's a phase 2B, and I think it's not as exposed to the US investor base. And you know, Uli, tell us what's happening over at Vivoryon.
Yeah, first of all, thanks very much for having me on the panel, and let me give you a very top-line overview what Vivoryon is about. Actually, we are a public European biotech company listed at the Euronext in Amsterdam, and we are targeting pathological post-translational modifications, which are important in the onset and progression of diseases. And initially, we are focusing on Alzheimer's disease, where we have discovered the pathological role of the enzyme glutaminyl cyclase and its isoform in 3 different aspects. So it's catalyzing the formation of a synaptotoxic variant of A beta called pyroglu A beta. Secondly, the isoglutaminyl cyclase is promoting neuroinflammation by stabilizing CCL2, and CCL2 as such is promoting the tau pathology. So by inhibiting glutaminyl cyclase and its isoform, we are modulating all 3 pathologies in Alzheimer's disease, which we feel is unique about our approach. Now, our lead candidate, varoglutamstat, is a small molecule oral inhibitor of glutaminyl cyclase and its isoform, and it's currently subject of a phase 2B trial in 250 Alzheimer patients in a European trial called VIVIAD, which is enrolling patients as we speak. And at the same time, we are preparing a phase 2 study in the US called VIVA-MIND, together with the Alzheimer's Disease Cooperative Study under the leadership of Howard Feldman, a renowned key opinion leader in Alzheimer's disease. Now, the study is supposed to start the second half of this year and is set to enroll 180 patients in a phase 2A, which is supported by an NIH funding, and a seamless stage gate into phase 2B with additional 414 patients. So overall, our comprehensive phase 2 strategy will consider almost 800 early stage patients and is supposed to deliver a well-balanced clinical readout with a focus on cognitive and functional parameters and based on treatment durations of up to 96 weeks. So we expect the full readout of the European trial VIVIAD in 2023 and VIVA-MIND in 2024, with an interim safety analysis presumably in 2022. Now, we at Vivoryon believe that we have a high degree of clinical validation for our approach with data from our phase 2A trial in 120 patients showing significant efficacy signals on 3 different levels: biomarkers in the cerebrospinal fluid, synaptic function measured by EEG, but also cognition. And additionally, recent data from Lilly with donanemab, which you mentioned in your introduction, is a further element of clinical validation since it underlines the relevance of pyroglu A-beta as synaptotoxic variant of A-beta. At Vivoryon, we have built a team with strong clinical development expertise and a unique network of partners in Europe and the US, such as Professor Philip Skeltons, the director of the Alzheimer's Centrum at University Medical Center in Amsterdam, our principal investigator for VIVIAD, the European trial, and the ADCS under the leadership of Howard Feldman, our principal investigator for VIVA-MIND in the US trial. And I guess I leave it with that for the time being.
Right, and I think Sharon and RJ and Remi, everybody's drug candidates, well, very different, are very similar in ways to Maria, where you span not just Alzheimer's, multiple neurodegenerative pathways can be impacted. So Sharon, tell us a little bit about why Cyclo has now expanded beyond Niemann-Pick into Alzheimer's.
Great, thanks, Jason. It's great to be here with you. So as Jason mentioned, our company has a lead asset on Niemann-Pick disease. And Niemann-Pick disease, as you may know, is a rare and fatal disease. Many patients suffer from cognitive decline. Now we are getting ready for our phase 3, first patient in with our Niemann-Pick study, so that is our priority. But at the same time, we know because of the similarities in the cognitive decline in both diseases that our drug has the potential to be a disease-modifying therapy. Our drug binds cholesterol. It has a particular affinity for cholesterol. And we know that Alzheimer's patients have an imbalance in cholesterol, which is why it's very interesting for us to use our drug in this indication. We know from animal data and from pathophysiology that cholesterol tends to accumulate in the membranes of aging brains in the hippocampus and the frontal cortex, and also from animal studies, we can see that cholesterol plays a major role in creation of some of the bad actors that we see, amyloid beta being one of them. There are animal data which show that given our drug, which is a cyclodextrin, to an NPC animal model, we can see clearance of amyloid plaque from the brain, reduce microgliosis, and so on. These are from the lab of Flint Beal at Cornell. We were asked to provide our drug based on this vast safety profile that we have from our Niemann-Pick program to provide our drug compassionately to an Alzheimer's patient, which we did for 18 months. And we were able to see indications of not only stability, the patient did not progress, but also improvement in word finding ability, less volatility, and so on. And so those are the data that we have taken to FDA now in our advice meeting and have the green light to submit our phase 2 IND at the end or in the second quarter of this year. So we're looking forward to getting that in. And we do believe that we have a possibility here to impact the amyloid beta and the tau through a unique mechanism that impacts cholesterol. I'll be happy to speak about that. But I'll add one other point. We do have a use patent pending for our drug, our Trapsol Cyclo drug for Alzheimer's. And we're looking forward to advancing our program strategically using that as another asset in the Alzheimer's program for our company. Thank you.
And RJ, TNF, right? That goes back all the way to the early 90s. And some of the work that you guys have been doing is so different from all of that hoopla around Enbrel. Remember that made its way into mainstream periodicals a couple of years ago. And what your company has done with MRI, you know, has been fantastic. And maybe you could talk a little bit about your dominant negative TNF inhibitor.
Yeah, so thanks, Jason. Most people don't realize that there are really two TNF cytokines. There's a soluble TNF, which is what everybody has been targeting to treat autoimmune disease. And then there's transmembrane TNF, which is really the good TNF, which is accidentally targeted by the current drugs on the market. And in neutralizing or actually in blocking transmembrane TNF function, you end up with all the safety side effects of long-term use of TNF inhibitors. And as it relates to the brain, that's mainly demyelination. In fact, chronic use of non-selective TNF inhibitors is now the most common cause of MS that is recognized. Most MS is idiopathic, but TNF inhibitors causes demyelination when used chronically. Our drug, using a very different dominant negative technology, only neutralizes soluble TNF. So we polarize that biology to transmembrane TNF, and not only do we get rid of inflammation, which improves microglial function as far as their garbage man role in the brain, which is critical, but we promote remyelination, we improve synaptic function, all in the brain. And we now have data from in man that shows all of this. And a lot of our data is really based on two sets of biomarkers. The first is the white matter analytics that Jason mentioned. Most people view Alzheimer's disease as a gray matter disease. They could not be more wrong. It is the pathology first arrives in the white matter. It is most easily measured in the white matter. And white matter analytics are remarkable, and we will be reporting some new biomarkers in August. But as far as measuring neuroinflammation, which is what we're kind of about, at least what part of our story is, clearly peripheral inflammation begets central inflammation. Central inflammation causes synaptic dysfunction and nerve cell death. If you get rid of central inflammation, you have a benefit, and we now have data from our phase 1 trial that the downstream benefits of getting rid of neuroinflammation include improvements in synaptic function and decrease in neurodegeneration. So we're pretty excited. We'll be starting a phase 2 this second half of this year. In fact, I had to step out of an advisory board to take this panel that we're having today that is helping design our phase 2 trial. So we're very excited. And like everyone on this panel, we believe two things. One, neuroinflammation is important. The whole game is not amyloid and tau. And most importantly, that the pathologies we are targeting have use beyond Alzheimer's disease in the CNS space. So these are exciting times.
And Remi, I saved you for last on purpose because I talk about all this activity in Alzheimer's disease, and what you guys showed in February at Cassava literally propelled the entire space upwards again. So maybe you could talk a little bit about your program and where things are with you guys.
Sure. Well, thank you. Thanks, Jason, and especially thank you, Maxim, for inviting us once again to your events. So Cassava Sciences is developing 2 product candidates, a proprietary drug candidate for the treatment of AD, and that goes by the unpronounceable name of Cemiphalam. That's the chemical name. We're also developing a blood-based investigational diagnostic to detect and perhaps stage Alzheimer's disease, and that's called SAVA. But the big excitement statement, I think, at Cassava Sciences is that Cemiphalam is phase 3 or will be phase 3 ready in the second half of this year following an end of phase 2 meeting with the FDA that was held in Q1. I'm going to give, for obvious reasons, I'll give you the abridged version of cimiflam, but the target of cimiflam is an altered form of a scaffolding protein called filament A. Filament A is it's widely known, widely accepted. It has a huge physiological role in the body, but that's not the filament A that we're targeting. Our drug only targets the altered form of filament A. Why? Because our scientists have discovered that the altered form of filament A is critical to amyloid beta toxicity. The mechanism of action itself is pretty complex, but enough to suffice to say that the altered form of filament A enables A beta 42 signaling via the very well-known alpha 7 nicotinic receptor, and also the less well-known TLR4. And by binding to the altered form of filament A, we restore the proper shape and function of the filament and curtail that link between A beta and A7. Bottom line is through this single target, we are able to reduce both neurodegeneration and neuroinflammation. That's a pretty big promise to make. Fortunately, we have replicated our results in phase 2. Most recently, we showed in a phase 2 randomized control trial, phase 2 study, we showed that cimiflam improved not one, but in fact an entire panel of biomarkers of disease pathology, biomarkers of neuroinflammation, and biomarkers of blood-brain barrier integrity. We also had an early test of cognition called CANTAD, and we saw very promising effects on cognition as well. Furthermore, we're conducting an open label study in patients with Alzheimer's disease, and we released we did a first interim analysis. We did a first interim analysis in 50 patients who completed 6 months of treatment, and consistent with previous data, we saw that in fact there was a beneficial treatment effect. Namely, cimiflam improved cognition and behavior in patients with Alzheimer's disease. ADAS-Cog scores, which is a measure of cognition, change in cognition to be more precise, we saw a 10% improvement from baseline to month 6. We also have a couple more interim analysis on deck in July of this year at AAIC. We intend to present our interim safety and cognition data in patients who've completed at least 9 months of open label treatment with cimiflam. And then in September, approximately September, we plan to announce similar results on safety and cognition in patients who've completed at least 12 months of open label. We also have a randomized controlled trial called the Cognition Maintenance Study that's ongoing. This study was initiated in May, so very recently. And like I said, the big goal at Cassava Sciences is to initiate our phase 3 program second half of this year.
So it's very important for everybody to understand what everybody here is doing. And now we need to get into how we ended up here. And one of those initial topics is how do we think about Alzheimer's disease in general from a drug development perspective? And Joanne had a very specific question to start us off with that.
So thank you all for those introductions. Just starting off from a more broader perspective, I wanted to discuss the overall change in view of Alzheimer's disease. Not so much viewing it as a chronic disease anymore, but more towards viewing it as a lethal disease, as it's currently the 6th most common cause of death in the US. So it would be great if we could discuss a little bit about the significance of this, and really anyone can chime in, but RJ, we'll start off with you if you could briefly touch on this topic.
Yeah, thank you, Joanne. You're absolutely right. It is the sixth most common cause of death in the United States, and we don't have a drug. That makes it unique. Turns out, not quite. Actually, the third or fourth most common cause of death is accidents. You can't have a drug for that. But every other one, whether it be cancer, cardiovascular disease, lower respiratory diseases, they all have a therapy. They have multiple therapies. They've had success in clinical development, and there has been no success in Alzheimer's disease. And I think both the academic and the biopharma community and the regulatory community are all partly guilty here. But I think what you see on this panel is a strong belief that if you bring thoughtful innovation based on science to the problem, there's opportunities to treat the disease. And no, not being stuck in the, I hate to beat a dead horse, in the, amyloid tau arena has allowed us, all these companies on the panel, to have some success. So I think the FDA and the public, well, the FDA is now viewing Alzheimer's as a disease that is killing patients and deserves some solutions, and understands that perfection is never the first step in any therapeutic area. So I'll stop there and let others chime in.
Is it perfection, Maria, or can we look at maybe getting drugs approved, and maybe even the one that's coming up with aducanumab, like oncology drugs? They start at the end, they get a real narrow label, they get good data, sickest of the sick, and then they work their way backwards. Is that where Alzheimer's disease and neurodegenerative diseases in general can head now, as we look at it today?
Yes and no. Yes for aducanumab, no for us. And the reason is, if you listen to all of us, we are all lowering inflammation and improving synaptic function, every single one of them. Well, if nerve cells survive, we survive. Because if our body doesn't hear, doesn't see, doesn't feel, we become vegetables and will die. And in fact, in the later stages of neurodegenerative diseases, all of them, so many nerve cells have died that we become vegetables and eventually die. We stop eating, we stop swallowing. So the fact that we can show, and in some of you have already shown it in humans, we have shown in animals, that we protect nerve cells from dying. We can actually count them. They don't die. And in this study that we are doing, the Alzheimer and Parkinson's, we haven't gotten to nerve cell death biomarkers yet, but I think we'll release them in the next month. So if we can protect nerve cells from dying, and a big part of that is lowering inflammation, then we do have a very different disease. One that we probably will prolong life. Now, I'm not really proposing to prolong life too much. We're going to die of something sooner or later. But that we can actually have a decent old age with dignity. And I think we can do that.
So Uli or
Jason,
yeah. Oh, go ahead, Remi.
Can I cut in?
Of course.
Yeah, so I guess I'm going to be the voice of caution here. The fact that there are no drugs to address Alzheimer's disease is really a large part of it is just due to the sheer complexity of the disease. There are so many things going on at the same time. And so I think we really need to approach Alzheimer's disease with a large degree of humility and perhaps caution on all of our data. On the one hand, it's an exciting time because finally the R&D field is shifting away from what predominated for 30 years or so. We're seeing the shift away from the anti-amyloid and perhaps anti-tau approaches. But I think as kind of the new generation of innovation, we do have to be very cautious in perhaps any overly optimistic interpretation of any of our data. It's a hard problem to solve, and it's going to take a lot of data. All data has to be replicated, and the data has to be rigorous. And without those criteria, I think we don't want to risk giving false hope to patients, or at some level, perhaps false hopes to investors as well.
But realistically, we'll all do phase 3 studies. So obviously the data has to be replicated in much bigger groups, and we'll prove it. The fact is that our preliminary data shows a lot more hope than the A-beta data.
And
Well,
just
I'll
a sort of,
chime
oh,
in.
sorry, yes.
Oh yeah,
Of course.
thanks. You know, I agree with you on the need for evidence, and I can hear from this group. This is an evidence-based group, evidence results and evidence-driven. I absolutely agree with the sentiment that this is a complex disease, whether it's tackling neuroinflammation or glucose metabolism, which we haven't talked about, or amyloid beta or tau or other features of the disease. There are 100 genes plus involved in this disease. I think that there are going to be many options. And that's what we're about, creating options for patients who are suffering. And so I'm actually very encouraged. I'm certainly encouraged about our own work, but I'm encouraged by the conversation as well.
Joanne, go ahead.
Yes, and just sort of remaining on, you know, the changes that's occurred within the last decade or so. I'd just like to briefly discuss what's changed, and in terms of the knowledge we have about the disease, including the implication of neurotoxicity and reversing the neurotoxic cascade. Really, anyone can chime in, but Maria, I think this question would be great if you could start it off.
Well, we were kind of lucky maybe, or we just had the right advisor. We had data we couldn't quite explain. And Bill Mobley said it's axonal transport. And I said, what? So he turns out to be an expert in how information moves through the arc of a nerve cell. And by having axonal transport in our cascade, we really were able to link the beginning of where neurotoxicity starts to the end where nerve cells die. And we have really beautiful data on how our drug improves the information highway and restores the information highway. And the reason I really feel very blessed that we ran into this axonal transport is it explains our data, it explains our animal data, and we were able to show it, and now we want to show it in humans. But it is so simple to understand that if our brain doesn't communicate with us, be it other nerve cells, be it the periphery, be it internal organs, we don't function. And in a lot of ways, I feel that that may be one of the most important discoveries in how to treat a neurodegenerative disease, because in every single neurodegenerative disease ever tested, axonal transport is impaired. The information highway is slowed. Now, that doesn't mean, and in fact, some of you companies that have inflammation, if you go back, you'll see axonal transport. It's not something that can be attacked just from one angle, because as Sharon said, we have so many different things that happen, and we can attack the same things from so many different angles. But I really feel that axonal transport put the diseases in perspective for us.
But Uli, that leads right into what Vivoryon is doing. You're also doing it with an oral medication like some of the other groups here, but you're disrupting post-translational modification. So you tie into altering synaptic transmission as well, or improving it rather. Right.
But just, you know, let me go back to the comment Remi was making, and I think that's an important one, because what we also have witnessed in the recent past is that we have seen nice anecdotal early data, which, you know, couldn't be validated in larger clinical settings. And my message is also that it's important to have, you know, the right clinical setting and the right clinical development strategy. That's also a success factor in developing drugs. And you know, what has changed in today, and what makes me very hopeful is, you know, 10 years ago, most of the approaches were kind of, you know, pursuing this beta hypothesis. And what we know today is obviously the plaque load in the brain does not correlate with disease progression. I think that's been pretty clear. And why do I believe we are at a kind of tipping point? Is that now we see a diverse bucket of approaches which will, you know, generate much more knowledge in the field than it has been in the past, with this kind of very strong focus on the I-beta hypothesis. I mean, there was a reason for it, and it cannot be ignored that, you know, from a genetic perspective, there's a lot of evidence that the A-beta metabolism will play a role, but I think the consensus is there. It's a complex and multifunctional disease, and yeah, again, our approach is kind of, you know, addressing multiple pathways and multiple pathologies, and of course, yeah, we are focused on retaining synaptic function.
So obviously the big thing that's happening is going to be on June 7th, right? Everybody's watching. I'm not asking anybody to handicap and try to pick what, you know, what the outcome could be, but here's a more broad question to maybe get around that, is how does the FDA handle this? Does the FDA go against its outcome? Does it need this drug approved, at least on some level, to invigorate the space, give patients something, it's relatively safe, and then kind of go from there? Or does it just walk away from aducanumab? And RJ, we've talked about this at length, so I'll start with you.
I think it's pretty simple. If this was an oncology drug that gave you a 32% improvement in outcome, there would be absolutely no question that they would approve it, right? With all the warts and pimples, they may give it a conditional approval. They may make for post-approval commitments, but a 32% benefit is not, singing in the winds. So I think they're going to have a very hard time saying no, despite all the chatter. I do agree, and I've said this from the beginning, that they're going to put it in a tight little box. You know, they're going to say, okay, you know, you need to do it again with a post-approval commitment. I think they're going to ask them to try to understand or fix the safety problem with ARIA. But the bottom line is, I think you've got to, as I said before, you have the sixth most common cause of death in the United States and no drugs. Here they have one, they can start the journey. So I'm quite optimistic, and I benefit, and above all, I think patients and their caregivers are going to benefit.
Right. I wanted to ask, right. Oh, sorry. Yes.
Go ahead.
Okay. No, I just wanted to ask this question about can aducanumab and donanemab can they be interchangeable? Do you think that we'll end up with many anti-amyloid drugs? Like now we have 7 immune checkpoint inhibitors.
It's a good question, right? Because Lilly's not giving up, right? After solanezumab, they're right back at it with another one. Is Sharon or Remi, is this where we're heading with amyloid and antibodies? Are they just going to be here as a part of the treatment paradigm? At some point, they're going to be approved.
Well, let me say one thing about and comment on RJ's response. If there's anything that works for the patient community, then that's a win for everybody, because these patients are desperate, so are their families. So one can only imagine that companies would continue on as aducanumab and others on the monoclonal antibody side. I do want to say, from our side, with cholesterol, which we believe can have an impact on amyloid beta. And how can it do that? We have to think about what cholesterol does in membranes. And Maria, this gets to synapses as well. Cholesterol is associated with lipid rafts and can anchor proteins within the membrane, including the enzymes which create amyloid, which is why we're very excited about this as a potential way to target amyloid. So I think amyloid is here to stay, certainly from our perspective, from that end. But also, Maria, to your point about synapses, there may be other ways that cholesterol is having an impact at the synaptic level, and so that's another area of going forward. but I'll yield the rest of my time here so that folks can comment specifically more on your questions.
Remi, any thoughts?
Around Biogen?
Just around plaque clearing. And are we going to have a whole bunch of these drugs at some point?
I mean, look, it's
to start.
You know, at the end of the day, the FDA is going to do whatever they do, and you know, none of us can know. None of us are the oracle of FDA. Having said that, it is a hypothesis that you know, it's a very, very strong academic hypothesis for what's going on in AD. So in that sense, it holds up very well. The problem is that that hypothesis is not translated in the clinic. I mean, how many more failures do you need before you reach no? I think we're up to 21 large phase 3 trials that have failed. It'll be interesting to see what happens in early June. I think the real question is what might be the impact for, call it the innovation crowd, which is all of us and several more, who are approaching Alzheimer's disease with a very different, from a very different data perspective. You know, perhaps on a selfish level, I am rooting for an approval. And by this, I mean that investors, I mean, clearly patients and physicians need a win, but you know what? Investors need a win as well. Too much money has been poured down the anti-amyloid drain, and it's still circling. Investors really need a win, and at some level, FDA needs to show to the world, and the world at large, all stakeholders, that okay, even though this may be a stopgap measure, we're really prepped for something to address the problem of Alzheimer's disease.
I wanted to discuss the advances.
Oh, Uli, go ahead. Sorry.
Oh, sorry.
Yeah, I mean, just you know, one view from my side. I mean, so first of all, so I don't think that, you know, this PDUFA date is going to be a make or break moment for the entire field. And you know, in our minds at Vivoryon, this date is a decision point which is not binary, and it's what it is. It's going to be a specific decision on a particular data package presented by Biogen, which has been even controversially discussed, obviously also within Biogen. And you know, I resist the temptation to make any prediction how that will play out. But for us here at Vivoryon, you know, the data readout of Lilly's donanemab is a much more important, you know, element of external validation. But having said that, of course, I mean, overall, it is important for all of us in the field to learn from each other, whether it's from successes or from challenges or setbacks. And ultimately, we are all working on, you know, towards improving the lives of patients and their families. And any progress towards approved therapies means a step forward for all of us, I guess.
Right, that's definitely, oh, sorry, but that's the right thought, right, Uli? Because you're concerned about the patients, and it is just a moment in drug development without a candidate, for better or worse, whatever happens. But I look, and I don't really have opinions on here, but I do this time. You know, we've been following the space for years, and if it gets a CRL, you know, where does the investment community now look? Is it shift? I mean, you guys have been building traction for the last 2 years. Now does it entirely shift away from the antibodies and now more towards being even more open-minded about all the neuroinflammation and things like that? That's what we're looking at from the analyst side. But Maria, I'm sorry, I jumped in and cut you off there.
I wanted to second what Uli said. I don't think it's going to have a huge impact on all of us, whether it gets approved or not. And in terms of having a whole bunch of antibodies out there, maybe I kind of doubt it. The effect is not so huge. If you start thinking of combining antibodies, you have too many side effects. Most of, actually all of our drugs are all orally available small molecules. We can think of having combination therapies. That's hard to do with an antibody. So what, you know, somebody else said the FDA is going to do whatever the FDA is going to do. They may decide that patients do need something. And I agree, patients do need something, but we have some data here around the table that show an improvement over baseline. Biogen shows a huge decrease over baseline. So yes, it's better than placebo. I'm not arguing that. So the FDA is going to do what the FDA does, and we're going to continue developing our drugs. And one of these days, we're going to look at putting them together, because every single one of them has a different mechanism, which means they could be synergistic. And that's really what's so encouraging.
I believe anybody that thinks that the decision by the FDA won't have an impact on our companies is whistling in the wind. It is going to have a huge impact. If it doesn't get approved, investment in the space is going to be more difficult, full stop. If it does get approved, investment in the space is going to be easier. And as you know, other than IP, cash or capital is the lifeblood of this business. And the harder it is to get capital, the harder it is to do your drug development. So I'll go out on a limb here. I think that this is a watershed moment. And I think the FDA is not blind to that fact. And to believe that the FDA is not a political animal, just look at the HIV epidemic of 2 decades ago, the way they responded to public pressure. So, I come down firmly on the side that this is a big day coming up. So,
well, not to make this about capital and things like that, but I'm pretty sure just generally the companies on the space are capitalized sufficiently for at least the near future. And I'll just leave that there. So that's a positive, and we'll see what happens after Aducanumab. But Joanne was going to take us down a little bit of a different road with biomarker utility.
Yeah. Hey, Jason, Can I address this last point?
Of course.
Before we switch topics? Yeah, I mean, I generally agree that in the short term, there will be a lot of noise, whether it's thumbs up or thumbs down. It will have an impact on all of us in the short term. But I think once the noise clears away, the one thing I do fear about an approval is that every big pharma and his cousin will now dust off the shelf all of these anti-amyloid antibodies that barely worked 2, 3, 4, 10 years ago, and go run to the FDA and say, "Hey, me too. You gave Biogen an approval. Where's mine?" I think that would have an even greater impact on us as smaller innovation companies for a number of reasons. One is that we can't compete with big pharma unless we generate lots and lots of data. But more importantly, from a tactical point of view, there are only so many, there's a limited pool of patients. There may be 5 million patients in the US with Alzheimer's disease, but we each have entry we each have entry criteria, and not all, in fact, very few patients qualified to enter our trials. So I guess I might be a little more concerned about an approval for Biogen followed by a rush to FDA with a dozen other so-so antibodies.
Well, now before we change topics, let me kind of add a little bit more to your point. And I think Joanne mentioned, are we going to have all of these antibodies emerging if they get an approval? And then to your point, Remi, we're going to have a whole bunch like PD-1 inhibitors, checkpoint inhibitors in oncology, but that wasn't such a bad thing because now they know that those on their own are not good enough, but you can combine them, and everybody is trying combinations in oncology. There's thousands of trials, and the owners of those PD-1s, the big pharmas, are partnering with everybody trying to get those. Can we do that in Alzheimer's disease as well? Maybe that's not such a bad thing, having 8 plaque clearing antibodies out there. Maybe that's what you need as a stepping stone for combination therapies. Anybody want to take, Uli, any thoughts on that?
Yeah, I mean, I kind of like the analogy to oncology, because if you look on the prevalence of all cancers, it compares to the prevalence of dementia, of which 60% is Alzheimer's disease. And, if you look on the diversity of approaches in oncology and, the investments going into that field, I don't think that we in our field are at a comparable, point as compared to the oncology development. And I absolutely agree. I mean, if one drug makes it, and then it's kind of a starting point for various combinations, which are a kind of logic approach in a multifactorial disease as Alzheimer's disease. Absolutely.
Sharon, is it outrageous to think like, oh, we'll pick any of the drugs that you got, oral drugs or subcutaneous in Immune's case, or even an IV of Trapsil Cyclo, if you're getting your aducanumab or whatever plaque clearing drug, you now need to go in and get some of your cholesterol kind of flushed out, for lack of a better phrase. It's possible for a combination, no?
Yeah, in fact, we anticipated this in our patent, our use patent, which is pending, as I said, combination therapy. And our drug is given intravenously over slow infusion, hours. And so, Jason, we're 100% aligned with that thinking. Giving a drug IV to manage the cholesterol-related aspects of Alzheimer's, coupled with potential other, makes great sense.
Joanne, did you have a comment?
Right. Yeah, just retracting a little bit, just on the topic of shifting away from antibodies. There's been advances in the utility of biomarkers, which have really evolved in the past 2 years. So maybe Remi, you could start us off by touching on your ongoing trial, which incorporates biomarker assessment, and Uli, yours as well with the phase 2 trial.
Sure. So if you think about what is Alzheimer's disease, it's clearly a disease of biomarkers. And in that regard, we are heavily biomarker driven, and our phase 3 program is heavily biomarker driven. Which biomarkers? That's always the $1 billion question. Which biomarkers are more important than others? Hard to say. However, what the literature does speak to is the so-called core biomarkers, which is A-beta 42, total tau, and p-tau. I think for most researchers, those three gauges are probably what counts the most. And in fact, p-tau 181, either 181 or 217, I forget. One of the two is being validated as a diagnostic for Alzheimer's disease. So I would say those three biomarkers are probably universally accepted as diagnostic criteria for Alzheimer's disease. Beyond that, perhaps at a second level, then you've got all the biomarkers of inflammation, biomarkers of pathology, of neurodegeneration, which are also very important. And in fact, so important that a number of companies are now working on, bigger companies are now working on, once again, antibodies against things like TREM2. It gets a little harder to comment on kind of the second level of biomarkers, other than to say what is well known is that in general, as these biomarkers go in the up direction, cognition goes in the down direction. And I think what we're all betting on is the reverse of that, which is to say that if we can suppress these biomarkers, then we have a shot at improving or at least stabilizing cognition over the long term.
A new emerging biomarker, maybe it applies to Cyclo more specifically around cholesterol metabolism coming. Since your drug does this, clears cholesterol out into the bloodstream from the brain, you could see it, right, with the 24S marker that can you talk a little bit about that?
Yeah.
Maybe it finds its way into Alzheimer's disease now.
Yeah, thanks. And we have some data from our Niemann-Pick program, which has a cognitive decline element, which shows that when we give our drug intravenously, we can indeed affect biomarkers from the CNS, and that includes tau. So tau tends to go down in Niemann-Pick patients over time, very interesting, but also very importantly, because our drug crosses the blood-brain barrier, we can see that 24S hydroxycholesterol, one of the metabolites of cholesterol, which is exported out of the brain, actually increases in CSF and in serum. We measure it in serum. So we're seeing a direct effect of our drug on the CNS after we give it intravenously. And then again, that tells us that if we're having this impact on cholesterol, excess cholesterol in the brain, in these aging brains, then we can potentially have a disease-modifying therapy with respect to those proteins whose function depends on proper amounts of cholesterol anchoring them in membranes. So we're very excited about this as we move into our phase 2.
Oh, that's great. So Uli, what are your thoughts on biomarkers and specifically your drug, which has an impact on them as well? Can they be correlated to cognition changes?
I mean, that's the ultimate goal, and we are still in a kind of exploratory phase here. But of course, biomarkers are very important for the disease because they are defining the diagnosis. Yeah, the diagnosis of Alzheimer's is based on, the proportion or the relation between A beta and phosphorylated tau. So they can't be ignored. And for us, it's important to have a proper diagnosis as inclusion criteria for our patients. And then, of course, we are looking on different levels of biomarkers. One important one is target engagement. So we want to understand what is the level of target occupancy, and we follow that in the cerebrospinal fluid, as well as inflammatory biomarkers like YKL-40 and also markers reflecting synaptic integrity. And what is also important in our development strategy is that we also have a partner on board who is a world leader in the development of blood-based biomarkers, which is Nordic Bioscience. And we are also looking into those, and that what I meant is a kind of exploratory element, because the expectation would be that we can correlate those blood-based biomarkers, which are which can be acquired on a much less invasive procedure than the lumbar puncture, would correlate to, for example, cognitive readouts or even functional readouts. So for us, the biomarkers are a very integral part of our development strategy.
And Maria, can you expand a little on the correlation between a biomarker and the neurotoxic cascade?
Yes, absolutely. I agree that while the most important or the most known biomarkers are the A beta and the tau ones, I think what we're here to say is that if just A beta is affected or just tau, maybe we don't have an outcome. So we really need to show what happens after we reduce A beta, or in this case, after A beta goes up and tau goes down, and phospho-tau, after we have affected the most known markers, we really need to show what happens next. In our case, we're actually looking at an increase in axonal transport, a decrease in inflammation, which we have shown so far, and then a decrease in synaptic markers. And if we have that in Alzheimer's and in Parkinson's, I feel very strongly we should have efficacy. Somehow we have put the cart in front of the horse. We have efficacy, and now we're doing biomarkers. I really feel that if we can show a process, how the brain goes from A to B to C to D, then we show how our drug acts. And that is much more important than showing one very specific biomarker and assuming that's good enough.
Well, something also very unique, I think, emerging, right, RJ, is what you guys are doing with MRI as a potential biomarker. Maybe you could talk a little bit about that. And Maria, you'd mentioned cognition. After RJ, I just want to bounce it over to how these things translate into cognition. This is exactly what we saw with Cassava's drug in February. But RJ, maybe can you just talk a little bit about the MRI approach?
Yeah, so thanks. And let me start with a comment. I don't get amyloid. 40% of cognitively normal elders have a boatload of amyloid, right? And people have amyloid 20 years before they develop any dementia. So by any other standard, amyloid would not be a biomarker of disease, but it has legacy status and was grandfathered in for Alzheimer's disease. So I don't get tau, I'll accept, but I don't get amyloid. Now, our approach is, you know, I'm not dumb. I look at where do all the drugs get approved? They get approved in oncology. Why does oncology get 10 new drugs approved a year and CNS gets 1 new drug approved a decade? It's because they use biomarkers. And how do they use biomarkers? They use it for patient selection to make sure that they align the disease that the patient has with the mechanism of action of their drug, because they've assumed that all cancers are the same. I already mentioned breast cancer and leukemia. So that's what we use our biomarkers for. Our biomarkers, we've made the assumption, as has been said multiple times here, that Alzheimer's disease is a complex disease. The term Alzheimer's disease was generated, 115 years ago. And the assumption that has been made by biopharma is it's one disease, and you and I know it is not. So I'm not going to treat a disease that has primarily a cholesterol synthesis problem or primarily a glucose-related metabolic problem with an anti-inflammatory. I'm going to treat a disease where inflammation is causing the dementia with a drug that targets like XPro 1595. So that's what we use our biomarkers for. And we have found 2 things. Peripheral biomarkers of inflammation get you started, but their precision for predicting what's happening with neuroinflammation is not perfect. And secondly, patients don't, it's hard to do clinical trials if everybody has to get CSF, has to get an LP, right? So we've decided that there are some really interesting emerging technologies with imaging that are non-invasive, where you can look at white matter pathology and gray matter pathology. And we think that white matter pathology is where we should start, as I said before, because it predates gray matter pathology. It's more defining. And so, in fact, in our phase 2 trial, patients, we know that we aren't going to guess that patients have neuroinflammation. We're going to know they have neuroinflammation associated with their Alzheimer's related dementia, okay? Because at the end of the day, I don't want to be treating a patient that has a variant of Niemann-Pick disease, basically. Not, Sharon, I'm not being critical here, but I'm just using it as an example, a disease where cholesterol synthesis is the primary pathology, because I can't fix that. So what? I only treat 30% of the patients maybe, but I'm very happy with 30% because we all know as small companies, we don't get rewarded for the size of the market we get a drug approved for. We get awarded for getting a drug approved. We can expand the market later, and that's what our plan is. So we believe that biomarkers are essential to select patients, to drive success, and that's how we build our clinical programs. And really, we've just taken a page out of the oncology playbook that has been so darn effective for the last 2 decades.
But to Joanne's point is that they've evolved over the last 2 years. And I'm going to bring it back to Remi because it was his company's phase 2B where the biomarker data looked fantastic, and that was last September. And it actually translated into cognition improvements all the way out to 6 months. And we could discuss either side of that, as many do, but how are biomarkers now moving into mainstream utility, Remi, and even potentially shortening trials?
Shortening trials, I don't see that. Again, this is a very complex disease, as we've repeated many times. So I don't see anything that will shorten trials. On the other hand, yes, I think let's step back a second. The FDA requirement for approval is to show subjective improvements on both cognition and health function, okay? For better or for worse, it's a co-primary endpoint that they want. Hopefully things change in the future, but for now we're all stuck with the necessity to improve over time, both cognition and health function. Now, cognition is relatively subjective. ADAS-Cog can fluctuate by a few points that, from overnight, from week to week, from month to month. This is why you really need a lot of patients and multiple time points in order to get a truly validated sense of how a drug impacts cognition. I think in our case, whatever acceptance we have, Cassava Sciences has, is not necessarily because of the phase 2b results, but rather because the phase 2b results replicate an earlier study. And furthermore, the earlier replicated study replicates the animal the animal data. So there's kind of consistency across the board. And that's what I think investors really want to see, is they want to see replication and they want to see consistency all the way from basic biology to mechanism of action to biomarkers and ultimately to cognition.
And we could talk about this for probably hours. And we're up against time, and obviously Alzheimer's, as we started this talk or panel, it's going to be busy. I think I'm with RJ. I don't think there's any way everybody's not impacted by what happens on June 7th, which is why we wanted to get you in front of everybody now to see what's going on outside of aducanumab. And we're also looking forward to AAIC and all the activity around Alzheimer's this summer. So lots cooking in Alzheimer's disease. But I would ask each of you to leave us or viewers with, what are the next steps or events around your Alzheimer's drug candidates, over the next 6 to 12 months that they could look forward to. And I will start with you, Uli.
Yeah, well, as I mentioned, there's a lot of things ongoing within our pipeline, and the next important event will be the start of the US trial, and of course, what I have mentioned also in my introduction is the safety assessment in our European trial based on the safety and tolerability data on the first 90 patients. And on the other hand, and we all know that, developing drugs in Alzheimer's disease is a kind of marathon. So to get the full picture of the readout of our trials will take until 2023. That's kind of a fact. But we believe that the data readout of our very comprehensive phase 2 strategy really has the potential of a massive value inflection for our company.
Sharon, no particular order. I'm just going around the horn, as they say.
Okay, great, thanks. And I'll just summarize some of the points that I've made so far. So next up for us is the second half of this year. We will submit our IND for phase 2 study to FDA. Looking forward to that. And remember, we're allowed to go to a phase 2 and skip a phase 1 because of the vast amount of safety data that we have on our drug from Niemann-Pick, including from some patients who are in their 60s. So we're very excited about that. And then the last point is that we do have our patent pending, our use patent. And so we're going to use that and all the data that we have at our disposal as we work strategically to advance our program forward. So thank you.
And I know we're Alzheimer's disease, but Sharon, I'll just do it for you. Cyclo also have another big event in their phase 3, and Niemann-Pick is coming up. And there's a PDUFA out there related to Niemann-Pick that we're watching very closely in June as well. So we'll be looking for that. RJ, just give us a kind of a recap on what we can.
Simple. We have additional biomarker and efficacy data from our phase 1 trial, which is small. But really, it's all about announcing what our phase 2 design and enrolling those first patients in the fourth quarter. So, at the end of the day, the rubber meets the road at a adequately powered, blinded, placebo-controlled trial.
Maria, I'm looking at you in front of me.
So we had reported the data from the first 28 patients of the study. We have another over 40 patients to go. So to round out the whole study, it will be 68 to 70 patients. We expect the data from the additional patients in the next 3 to 4 months. then we will ask for a end of phase 2 meeting with the FDA, and hopefully we can start one study in Alzheimer's disease and one in Parkinson's disease in winter. And we hope they're phase 3 studies, but that will depend on the FDA. So 2 phase 3s.
And Remi, if you could please finish us off with any upcoming events for Cassava, specifically the 9-month and 12-month data that we're looking forward to.
Sure. So I guess the parting words of wisdom would be what I would hope for is to see both replication and consistency of data. Our field is data-driven. It has to be. It has to remain data-driven. So again, I would kind of insist on replication and consistency across the board, and also kind of full disclosure of all available data, the good, the bad, the ugly. As far as Cassava Sciences itself, we're very excited about presenting at the upcoming AAIC conference. We were allowed an oral presentation of our interim analysis, 9-month interim analysis of the ongoing open label study, where we intend to present both safety and cognition data. And then later, a few months later, we intend to announce results of the 12-month interim analysis, again on safety and cognition. And then, of course, the big deal here is the goal to initiate a phase 3 program in second half of this year.
Okay, and with that, I want to thank all of you for participating, and to all the viewers. It's June 7th, the date. That's what we're all watching. We're also watching AAIC, and if you think about 2021, Alzheimer's is one of those topics. It's a hot topic this year. We expect it to continue to be, and we'll see what happens on June 7th. But now everybody can go start thinking about Memorial Day. We needed everybody thinking about what's going on outside of aducanumab. I think we've done that, and we look forward to seeing you again on our next MVest event. Thank you.